Hi guys, this is Randy Tang with my group, Kyle Manny, Jasmine Soni, and Nikhil Kumar, and this is our presentation about mitosis and the life cycle of a cell.
This is the endomembrane system, and it consists of all the organelles that are suspended inside of it. And the organelles are the nucleus, the ribosomes, the endoplasmic reticulum, the Golgi apparatus, the vesicles, the lysosomes, and the plasma membrane.
The first step of the life cycle is interphase, and it's the longest stage of the cell life cycle, and it takes up about 90% of it. The interphase is made up of three phases. The first one being G1, in which the cell grows and grows necessary organelles such as the mitochondria, the endoplasmic reticulum, the Golgi apparatus, and other organelles. When the cell senses that it is ready and the environment is suitable, it moves on to the next phase. Mitosis is the second part of interphase where the cell replicates its DNA for the second nucleus. The second copy of DNA is stored for the second nucleus. G2, the last stage of interphase, is where the cell undergoes rapid growth and it synthesizes the necessary proteins, such as tubulin, actin, and myosin, for the next part of the cell's life cycle, which is mitosis.
Okay, so the next stage in the cell's life cycle is mitosis. This is when the cell's nucleus split into two daughter nuclei. The first stage of mitosis is the prophase. This is when the nuclear envelope surrounding the nucleus breaks down and the chromatin inside condenses into chromosomes. Also, in this stage, the centrioles move to the poles of the cell. The next phase of mitosis is metaphase. This is when the spindle fibers attach to the centrioles, they connect to the kinetochores in the center of the chromosomes. Then they align the chromosomes along the middle of the cell to form a metaphase plate. Anaphase is the third stage of mitosis. This is when the spindle fibers start pulling the chromosomes apart. When the chromosomes are pulled apart, they become two sister chromatids, which are then pulled to the opposite ends of the cell. The last stage of mitosis is the telophase. Basically, this stage is the opposite of the prophase, and this is when the nuclear envelope reforms around the nuclear material in the opposite ends of the cell. This forms two daughter nuclei. The last part of a cell's life is cytokinesis. This is when the cell splits into two daughter cells. This is done by microfilaments wrapping around the equator of the cell and then squeezing. This forms a cleavage furrow, which is where the cell will eventually split. While it is squeezing, the organelles inside of the cell, they basically move to either side of the cell. They divide pretty much evenly. The Organelles like the Golgi apparatus and the endoplasmic reticulum, they kind of split in half, and half of them go to each side. When the microfilaments fully contract, the plasma membrane collapses on itself, and then the cell splits to form two daughter cells. And then the cell life cycle begins.
So, the endomembrane system also plays a large role in the life cycle of the cell. It produces many proteins that are integral to mitosis. For example, we will talk about how microtubules are made inside a cell. First, some of the DNA inside the nucleus is transcribed into messenger RNA. The messenger RNA is transported in a vesicle to the ribosomes on the rough endoplasmic reticulum. Subunits of these ribosomes are uses transport RNA to bring amino acids to the ribosome and fuse them together. This step is called translation. Then, this incomplete ribosome is transported to the Golgi apparatus by a vesicle. There, the Golgi apparatus manipulates the protein until it passes through all the cisternae. The final product is the Golgi apparatus. The tubulin is put together to form microtubules, which are transported by vesicles to form spindle fibers in the cytoskeleton of the cell. Spindle fibers play an important role in mitosis. They pull apart the chromosomes during anaphase and separate the sister chromatids and also pull them to the poles of the cell so that during telophase, two different nucleus will form.